Hello, everyone. This is Chapter 13, Regulation of Gene Expression. So this is a chapter that we haven't gone over in your ninth and 10th grade biology classes. This specifically looks at how do we regulate that gene expression? How do we, tell certain genes to activate and tell other genes not to activate or deactivate? How do we have certain proteins being produced in some cells and not other cells? How do we have these different types of cells? So this is where your body is going to really differentiate the number of genes that you have in your body. You have around 20 to 25,000 genes, and not every gene is going to be activated in every single cell. There's actually only going to be a small portion on within every cell that's going to be activated. So this is how your body and prokaryotes, those are those single-celled bacteria cells, how they are going to regulate their genes as well. Now, with this chapter, I'm going to go through in kind of different order. I have it here in the PowerPoint from the book, 13.1, 13.2, and 13.3. 13.1 looks over prokaryotic regulation, 13.2 eukaryotic, and then 13.3 is mutations. I am actually going to put 13.3 before 13.1, so I don't know where we're going to be at with the content here, but this section is 13.1, which might be the last or the second. I don't know what order I'm going to put it in because you can kind of put it anywhere. That being said, prokaryotic regulation, this wasn't really understood until the 1960s. Again, we're just figuring out DNA. We're figuring out that proteins are produced from the instructions on DNA. And we found out this kind of regulation first. And it's a little bit easier because we were looking at prokaryotic cells. We have something called a operon. Operons are a location on the chromosome where you're going to have a couple different sequences here. And this here shows you DNA, and every portion here has a little bit of a different region or name. We have our regulator, our promoter, our operator, and our genes. And each has a specific role within controlling gene expression. And by gene expression, what I'm saying is it's going to regulate how much of a gene is transcribed and then translated. So the promoter, this is a short sequence of DNA where the RNA polymerase binds to the DNA in order to start transcription. So you can see here the promoter sites. They're going to be like little locations on the DNA that say, hey, RNA polymerase bond here because the gene is next. So this is, again, just a region of DNA that RNA polymerase binds with. This is called a promoter. The regulator gene is found on the outside of the operator, or I'm sorry, the operon. This creates something called the repressor. So again, it's going to be transcribed and then translated, and then this protein will actually repress RNA polymerase from actually transcribing a gene. So think of this as another location, again, outside the operon, where you're going to have a gene that produces this repressor that stops transcription of a certain other gene. I know it's a lot of terminology here, but this is a great diagram so we can look at. Again, we have our promoter, and then outside of here is our regulator or a regulator gene. And this is going to be what kind of regulates this gene. So this is like a switch that can control this gene, if you want to say that. Remember, this is DNA that's going to go through transcription, that's going to go through translation and produce those proteins. Oh, yeah, we have some memes here. Yeah, the repressor stops the RNA polymerase from binding with the gene. Now, the operator, this is the location where the repressor is actually going to bind. And if you remember in that last image, it's on the inside usually of the promoter. And this usually either it stops RNA polymerase from binding, or it actually stops it from passing. You can do either one. Each operon is different. There are thousands of operons in nature. Each operon is different. We're going to go over the lac and trip operons in this section, but there's a lot of different types of examples of this. But the operator is just, again, a location on the DNA where the repressor is going to bind. The genes are going to be what actually is going to be transcribed and translated then to make a certain type of protein. So these are just regular genes that we've talked about before. Let me go back here. I'm going to show you this. So again, our promoter is where the RNA polymerase binds with. Our operator is where the repressor binds with. And remember, the repressor will either stop the RNA polymerase from binding with the promoter, or it will just stop it from getting past here and transcribing these genes. Again, we're trying to regulate these genes here. You can see here that repressor is not allowing the RNA polymerase to bind, and then it won't transcription won't occur. Tryptophan is going to be our first, or our trip operon is going to be our first operon that we're going to talk about. I have what you really need to know here. Again, it's pretty complex, but here are the basics. RNA polymerase binds with the promoter region. It produces an mRNA sequence, and that mRNA sequence goes to make enzymes that make tryptophan. So tryptophan is this amino acid that we're going to need in the body. Whenever you don't have enough tryptophan, I shouldn't say in the body, we're talking about prokaryotes, but whenever they need tryptophan, they're going to produce these enzymes that produce tryptophan then. And it makes sense. When I need tryptophan, I'm going to produce something that makes me tryptophan. So whenever this cell has enough tryptophan, though, what happens is the repressor is actually going to bond with the operator, and the repressor will actually stop the transcription of those genes. So you can see here, then the RNA polymerase won't be able to transcribe to make more of those genes. Here's why this is so important. This is like making cheeseburgers for lunch, like the lunch ladies making cheeseburgers. Say on a day where all the students are here, they need to make a lot of cheeseburgers, right? So the lunch ladies are all going to come in. They're going to say, hey, we need to make a bunch of cheeseburgers. Let's say there's a snow day. Well, there's no need for us to make cheeseburgers, so no one's here. So we're not going to have those lunch ladies come in. Let's say we have half the student body here. So it's one of those hybrid days. Again, we have half our student body. We might not bring in all of the lunch ladies to make all those cheeseburgers, because why keep on making cheeseburgers if we don't have the need for it? So think about regulation of how much things are produced, like let's say in the cafeteria. We're not going to produce 10,000 cheeseburgers, because obviously, even if every person eats 5 cheeseburgers, you're going to still have left over, and it's going to be a waste of energy, time, and cost. So think about cheeseburgers in these examples of operons. Now, when the tryptophan is used up, the repressor will actually change shape and unbind with the operator, allowing RNA polymerase to work again. You can see here we need tryptophan. Let's see, move this. There we go. We need tryptophan. So what's going to happen is you're going to have RNA polymerase bind here. It's going to transcribe the genes, make the enzymes that make tryptophan. So again, low tryptophan, RNA polymerase can pass. However, with enough tryptophan, tryptophan is actually going to bond with that operator, changing its shape, allowing it to bond to the operator, and then RNA polymerase can't bond because, again, we have enough tryptophan. The other operon we're going to talk about is the lac operon, or the lactose operon. So when the genes that we're going to specifically talk about is these genes that make the enzymes that break down lactose. And if you hear the word lactose, you should remember it's a sugar, it's a disaccharide, and these prokaryotic cells are going to have enzymes or genes that make enzymes that break down lactose. Now, whenever we have no lactose present, the repressor is going to stay binded to the operator. And it makes sense. If no lactose is present, why make enzymes to break it down? It would be, again, not efficient to make those enzymes. When lactose is present, lactose is going to bind with the repressor, which changes its shape. Once it changes the shape of the repressor will unbind with the operator, and then RNA polymerase can transcribe those genes, making the enzymes that break down lactose. Now, whenever this, again, I'm going to show you a little image here. Again, repressor is bonded here. RNA polymerase can't bind to the promoter, or it won't transcribe the genes. However, when there is lactose present, you can see here lactose, the repressor will unbind to the operator. The RNA polymerase can move past, making the enzymes that will break down lactose. Again, we don't need to break down lactose if it's not there, so why produce the enzymes if there's no lactose present? This helps the genes, or this helps the prokaryotes regulate the gene expression and how much gene expression there is. So again, if we don't have students here, why keep on bringing the lunch ladies in to make 100 cheeseburgers? They're all just going to go to waste. Now, there are two different types of these operons. We're going to call them repressible and inducible. Repressible is that it's usually activated unless the less the tryptophan or less the molecule there is present, then it's going to be turned off. Inducible means it's usually normally off, but then it can be turned, or it can be activated in the presence of that substrate, and that's the lac operon. So repressibles, again, they're usually repressed, and then it turns off. I'm sorry, they're usually repressed when there's enough of that substrate. Inducible is they're usually off until there is that substrate to break down.